Okay. So in class, we spoke about amino acid structure. And so from Laura's lectures, you guys know that amino acids, they all have at least the carbon in it. It's in the center of it. And then there's usually an R group of some kind, which is a special characteristic of which characterizes, excuse me, there is an R group that's involved in amino acids, and each R groups have special characteristics, and that's what makes each amino acid unique. And amino acids also have an amino group, which you see on the left. Maybe I can use the pointer. Can't see it. On the left here, you have the NH2, the amino group, and then the R group, like I said before. And then on the right side, you have the carboxyl group. And yeah, that's basically all you need to know for the amino acid structure. And amino acids based on pH, they're pH dependent ionized equilibrium. And yeah, I think that's it for amino acid structure. Does anyone have any questions so far? No? Okay. Chichi, do you want to do the next slide?
Yeah. So this is basically how peptide bonds are formed. So they're the water is formed as a product from the dehydration reaction. You can see. However, breaking the peptide bonds is a hydrolysis reaction. As you can see from here to here, that's the hydrolysis is occurring. I don't know if you can see my pointer. Can they see my pointer?
I don't see it.
Yeah.
No.
It's because you're sharing a screen, but
I
yeah.
have to give permission to click, I think, or something. Hold on.
It's okay. It's okay. And then when those peptide bonds are broken to release energy, and this takes a very, very long time to actually break.
Yeah, Chichi, if you click now, it will give you control of the screen. Yeah, and so we talked about polypeptide anatomy. Like Laura said today in class, you usually label your polypeptides from the N to C terminus, and you start numbering at the first amino group. And R groups are arranged by their chemistry and size. And peptide bonds, which is what holds amino acids together, they have resonance, which means for this class, resonance meaning that the bond has single and double bond characteristics. And yeah. Does anyone have any questions so far?
Can you repeat the part where you said they have, we can label the polypeptide from the N to the C terminus?
Yeah, so like when you have a polypeptide, it just is like labeling something from front to back. So it's just intuitively, like biologists label the polypeptide from the N terminus to C terminus. So kind of like in this image here, how like they count that like they count that there's like 4 different R groups, and so you start numbering from the left end or where you see the N terminus group. So there's like R group 1, 2, 3, and 4. So they're just showing you like how they would label a polypeptide with different R groups within it. Does that
Okay, thanks. Yeah.
Could you just go back one slide? Sorry, I just didn't get the last bullet point.
Sure. And I don't know if I said this, but after the review, I usually post this entire thing, like a recording on YouTube. So you guys will have that to see it as well. But yeah, I'll always be happy to go back so you guys can fill in your notes and stuff.
Thank you so much.
Of course. Any more questions? I'll take that as no. Chi, there's nothing in the chat.
The question is the prep for an assessment quiz? No, this is just an overview.
No, so the purpose of the undergraduate TA reviews are just to help you guys with topics that we cover each week. So I know it's misleading because the link I did for the Zoom, it wasn't my personal meeting room. I did the help sessions, so they're completely separate things. Like help sessions is more of a TA office hour that we have in the week. And then these review sessions on Wednesdays and Fridays are just sort of like for you guys' benefit, for you to rehear the material explained in our words. So yeah, these review sessions are not really like prep for assessment quizzes or anything. They're just to help you go over content that maybe you're confused about or that you'd want to hear again to review. Does that make sense? Okay. Yeah, is there another question in the chat? I just see like how many messages there are, but I can't see the message. No? Okay.
Okay, so there are four levels of protein structures. And the first level is the primary structure. So it's just kind of like the sequence of amino acids. It kind of starts from the N terminal to the C terminus. So it's held together by covalent bonds and the peptide bonds. Yeah, and really it's only by enzymatic activity protease that is. Do you want to add anything else, Michelle, or we're good?
Excuse me.
Mm-hmm.
What does the enzymatic activity protease do?
Okay.
It breaks down the protein
Yeah, so
structure.
you're saying that peptide bonds are covalent bonds, and for this class, like the covalent bonds are your strongest type of bonds. So that the bullet point there is just saying that the only way that you could possibly break a peptide bond is if you had a lot of energy, which is performed by that enzyme, the protease enzyme.
Okay, thanks.
Yeah, of course. And I wanted to go back. Somebody had a question about this slide here. I just wanted to go back and add a point here. So amino acids are labeled because they're synthesized from the N to the C terminus, and like how polypeptides are made, like proteins are synthesized from N to C terminus as well. So that's why the way that we count the R groups here, the way we read it is in order of how they're added. What's going on my screen here? Okay, Chi, I'm gonna like disable your control because
Yeah, it's being weird.
okay, abort control. Yeah, so as I was saying, the reason why it's sort of left to right N to C terminus is because that's how proteins are synthesized. And for this class, like proteins and polypeptide are interchangeable terms. But yeah, so how we read it, like how kind of like how in English we read left to right for reading an amino acid, you read from N terminus to C terminus. So hopefully that makes a little bit more sense there. Okay, and did anyone have any more questions about this slide here? No, okay. All right, and so second level of structure we looked at, or that are in the lecture videos on Moodle, is secondary structure. And secondary structure involves interactions between the backbone, and there are two main types of structures you see within the secondary structure, and these are alpha helices and beta plated sheets, or just beta sheets. And secondary structure, the most important kind of bond you'll see is hydrogen bonds. And yeah, so hydrogen bonds are pretty important for a secondary structure. They are involved in holding the backbone together. So the alpha helices kind of looks like sort of like a strand of DNA. If it had another helices with it, it would look like a double helix. So there are hydrogen bonds in there that hold the backbone together. So that's what I like to think of when I look at the secondary structure. And yeah, so like I said, hydrogen bonds hold together and hydrogen bonds are non-covalent. And without the alpha helices, excuse me, there are groups situated, or there are groups stick out on the outside, away from the helices, and the beta sheets, the R groups are interspersed up and down in the sheet. And the diagram here does a good job of showing that. You have the R group sticking up and down in the alternating fashion. And then in the alpha helices, you have the R group sticking outwards away from the helix, not inside the helix. Does anyone have any questions on that?
The secondary structure is just kind of chosen at random, right? Or is there anything that could like lead to an alpha helix being chosen or a beta sheet being chosen over the other, or is it just kind of random?
I'm not really too sure about that, but I know that she doesn't like delve into that detail for the purpose of class. But like some proteins have beta sheets, and then some proteins have alpha helices, and others have both. So I guess I'm not sure if it's random or not, but it depends on like different proteins.
Okay, thank you so much.
Yeah, of course. I do know, like at least from this class and like some other classes, that there are a variety of diseases you can get if you have like clumped beta sheets, like too many beta sheets. You can get like neurodegenerative diseases, and it's caused by prions. But that's like a whole separate topic that's beyond this that you don't really need to know about.
Okay, thank you so much.
Yeah, of course. Any more questions on this?
A question about the denaturation, or I don't know how you say it, but of like just proteins in general. So say like, I know you can have like, say you have a protein that doesn't have like the quaternary structure, but it has like tertiary, I'm trying to think. So if this is gonna come out really bad because I'm trying to like word my thoughts together. But so is that because like it won't affect, so when it doesn't affect another structure, so say it only takes away one structure, is that due to the interactions being different? So like if tertiary was taken off, but like secondary was still there, is that because of the different interactions they have? Because tertiaries are group interactions, and secondary involves the hydrogen bonds?
I sort of understand like where you're going with your question. I just am not sure if that's like a topic for this week, but we will get to that. Like we'll discuss, I believe, in a few weeks, maybe week 3, about how certain factors, like environmental factors, affect these bonds. That's why we're having you guys learn about the structure levels of proteins and the different bonds involved in each, because they are different in their characteristics and their strength. So, but I will give you an example just to see if it makes sense. But I don't want to lead you guys astray or confuse anyone. So I'm just going to try to think. So one of the factors we'll look at about that affects protein bonds, or like that can denature protein, is heat. So if you have a lot of heat acting on a protein, that can cause sort of the weaker bonds to wither away. And so that what would happen is like certain bonds that are weaker, like ionic bonds and non-covalent bonds, like all of your non-covalent bonds, which are your van der Waals, your ionic bonds, and some of the other ones that we'll look at, those bonds actually get affected by the heat, and it can cause like the protein to sort of become weaker. And then like covalent bonds that are very strong, like peptide bonds in primary structure, and if you have disulfide bridges, which are in tertiary structure, those bonds are very strong because covalent bonds are very strong. So those would remain intact. So like it depends on like the situation and what kind of environmental factor you have. And again, I know like that might not make any sense right now, but we will get to that topic in a few weeks. But you're right as to what you were saying, that each level of protein structure has different bonds that can be affected in different ways depending on like what would happen to the protein. So I don't know if that made sense or if that confused you.
Okay, thank you. I also just going along with that, just one last thing. So I know you said it has to do with like the strength of the bond. So does that necessarily mean that we can assume that like the strength of the bond like decreases, like primaries the most and then secondaries like quaternaries like the least? I'm just
It's not really a pattern like that. It's just more like the idea for this class is that your covalent bonds are usually going to be your strongest bonds, and for this class, like there's two covalent bonds mainly that you'll see, so which is peptide and disulfide bridges, and then the rest of the bonds like hydrogen bonds, van der Waal, ionic / electrostatic. Those bonds are considered to be non-covalent, and the non-covalent bonds are weaker than the covalent bonds.
okay,
So
thank
it's
you.
not so much like that there's like a sequence or order that quaternary is weaker and primary is stronger. It just has to do with like the bonds themselves. And we will like keep talking about this even on the Friday review, and I think probably next week, because this bond stuff is sort of like a key foundational idea to understand some of the other things that we'll talk about for the class. Did that make sense?
Yep, thank you.
Mm-hmm.
Can I just ask a kind of clarifying question?
Yep.
Kind of based off that one. So if like, I mean, I know you guys will probably go more in depth in like a couple of weeks and stuff, but so if like the secondary structure has it created like an alpha helix or something, right? And then later on, so if an R group came in and like the electronegativities were off or something, could that technically denature the secondary structure of the alpha helix initially? Like, could it change that? I don't know if that question made sense exactly, but could like R groups?
Denature is like, I don't think denature is like the right word to use.
Yeah, not denature. It could just like sorry, that wasn't the right word at all. It could just like change the initial structure of it. So like from an alpha helix or beta sheet, it could change that.
So I'm actually like not really sure how to answer that, but I guess like if you just think of like primary structure and how you have a sequence of amino acids that all string together, like a sequence meaning just like not like a list, but like just picture primary structure as like a string of beads, and like all the beads sort of connect together. And I think you'll go more depth into this in a few like of her practice problems, maybe this week or next week. But like if you had like a sequence of amino acids and they all had the same chemistry, so if they were all nonpolar, and then you removed one like in the middle and you switched it out for a group with a different chemistry, so if it was polar or like a polar positively charged, polar negatively charged, or polar uncharged, if you put a different group in there that has a different chemistry, then you have the potential to really mess up like the primary structure sequence would be different because of the order would be different, and you might mess up the bonds that are there. And like eventually like a protein would eventually, once the primary structure string of beads and you have like a long sequence of amino acids that continue to make secondary structure and then like tertiary structure, and eventually once the whole protein is made, you can kind of cause damage with that. And also in like tertiary structure, like if you tweak around different amino acids within there, or like if you switch different amino acids with different chemistry, it can have different effects. But it depends on like the situation of the question and stuff.
Okay, that makes sense. I was kind of thinking like once it gets to secondary to tertiary, I was like thinking like the R groups would start interacting like differently, and then that could like undo something. But I don't know if, yeah, I think that yours makes more sense. I think what you're just saying.
You're on the right track of thought. Like obviously, if you change something in a protein, like depending on what you're substituting it for, like there could be adverse effects. So I think like you're on the right track of thinking. It's just like sort of like more towards the future thinking. So I just don't want to confuse everyone else, but I see like what you mean, and depending on what's being disrupted or what you're what different amino acid groups there are. And depending on the size of the amino acid and the chemistry, there can be different effects. Chi-Chi, do you have anything to add to this? Because I'm like not too sure how to answer it.
No, you got it. Yeah, actually.
No, that makes a lot of sense. Thank you.
Yeah, of course. But yeah, so this is just secondary structure. We talked about primary structure, secondary structure. So secondary structure is when you have your alpha helices and beta sheets. And yeah, that's secondary structure. So primary structure is just like a string of beads, and then secondary structure is like when you have enough of those beads that form a shape. I guess you can think of it that way. And then, yeah, this is tertiary structure. I can't remember who was supposed to go, Chi-Chi. Was it me or you?
Okay, so tertiary structure is kind of just based on the organic chemistry, but it's kind of like it's generally stabilized by like outside polar hydrophilic hydrogen and ionic bonds, like their interactions. And then the inside of it is mostly hydrophobic interactions between like the non-polar amino acid side chains for that. And like the most special type of bond is like a covalent bond is the disulfide bond that you mentioned earlier when you were explaining. So it's kind of like an example would be like cysteine. I think we will talk about that in later classes. Cysteine is a type of disulfide bond. It has 2 sulfur bonds, and they're much stronger than any type of other bonds. So they really contribute to tertiary structure. So yeah, and pretty much all proteins have primary, secondary, and tertiary structures.
Yeah.
Any questions on that?
Yeah, most proteins have primary, secondary, tertiary, and not every protein has quaternary. Yeah, any questions on this stuff so far? Okay. And quaternary structure is separate from tertiary structure. And this slide just sort of outlines the difference between the two. So tertiary structure is when you have a single polypeptide chain and it sort of folds in on itself. And then quaternary structure is when you have 2 or more polypeptide chains that fold to create the 3D shape. And yeah, when you have multiple polypeptide chains, I believe that's called subunits. And for both of these structures, what they have in common is that they actually have the same bonds that hold them together. So they're stabilized by covalent and non-covalent bonds. And these types of bonds or R group interactions that occur are van der Waals, hydrogen bonds, electrostatic interactions, which is also called ionic bonds. And also some of them can have disulfide bridges too. And so, yeah, you can see in this diagram on the left, you have the tertiary structure. And then because the tertiary structure is just a single polypeptide or long protein chain. And then on the right side, let's see if I have the pointer. You have quaternary structure, which is 2 or more polypeptide chains. So you can see that the red is a polypeptide chain, and then the pink is also, it's tough to see with the colors, I'm sorry, this is a bad diagram, but the pink is also a polypeptide chain. And so she also goes over in lecture, I think she goes over hemoglobin. So hemoglobin, I think, had like 4 different colored subunits, so 4 chunks, like 4 different groups. So those are subunits and multiple, which are multiple polypeptide chains. Did that make sense? Does anyone have any questions on that? Okay. Yeah, so just like I said, the quaternary structure is 2 or more subunits and their interactions between the backbone and the R group. So yeah, this is hemoglobin, and you can see that it has the subunits here. And yeah, there are different colors to represent the different polypeptides.
If I can add something to the quaternary, just remember that each of the subunit in the quaternary structure has its own primary, secondary, and tertiary structure. So like there are 2 subunits or multiple polypeptides, but each of those have, like a polypeptide should, a primary, secondary, and tertiary structure. And also the quaternary structure, it's important because it's closely related to like its function. So like quaternary structures usually, it's like shape tells you what it's for. Like mostly they do biological processes like metabolism or signal transduction, which is something we're going to also talk about in this class. So quaternary structure is kind of like the highest, but also is very, very closely related to the function in the body. Anybody's confused or any questions, let us know.
I was gonna see if I could find like a video on YouTube to show of the structures, because I know it can be kind of tough to keep track of like what
I
different areas they are, what different.
I can look for it while you continue.
Okay. Yeah, I just
This might be, oh, sorry.
that's okay. You can go ahead.
This might be kind of like, I think that you kind of answered it, but so they each have their own primary, secondary, and tertiary structures within the quaternary structures. Well, like in their own subunits, they have their own like primary, secondary, and tertiary. So is that how you tell that they're like different subunits altogether and like that they have their own function is because of that? Or
So the way I look at this is like this is like hemoglobin. So hemoglobin is, I think it like carries oxygen to the bloodstream or something like that. So yeah, so this whole structure, like I think of this as like a mega protein, and like a mega protein can be made of like several small proteins or several small polypeptides. And so those small polypeptides that it's made of are subunits. So the subunits that this is made of, like there's, I believe because of the colors, it makes me think that there's there's 4 subunits, but like there's 4 subunits total, but there's like 2 types of them. Yeah, that there's 2 types of them, but there's 4 total. So this is a subunit, this is a subunit, subunit 3, and subunit 4. And so like within a subunit, it's like all of the subunits are like little proteins, and they come together and form this even bigger protein.
Yeah.
So I don't know if that made sense, Hannah. Did that make sense?
that made sense. I think I was just like confused on like, do they, when they like all the subunits together, are they also have covalent polypeptide bonds like bonded within the subunits? So like to each subunit, they're all bonded that way?
Yeah, so and like this picture doesn't really do a great job with showing it, but most of the pictures or a lot of the proteins you encounter, you can see like the different R groups or like the different N terminals or C terminals, and you can see that they're different proteins or different polypeptides, and then you can see with their interaction, like there's covalent bonding or there's like the hydrogen bonds, and you can see like the different types of bonding that goes on within the subunits. And then when you take a look like from like you step outside, how do I put this? Like you go deep into the mega protein and then you come right back out and then you kind of see it from a broad view. It's one big protein, but inside there's multiple interactions going on with different bondings. But
okay, that makes sense. Okay, thank you so much.
I have a question.
Yeah.
So the subunit is it the same way of saying protein domains for the subunit? Is it the same way? Is it the same thing?
Subunits and domains are 2 different things, and we're gonna explain that.
Okay.
They're different.
Okay, thanks.
Yeah. Okay, so no questions on this slide. No more questions. Okay. Sorry, Gigi, were you gonna say something?
And I was just looking for like
in a 3-letter or a 1-letter form.
So I'm gonna talk about the ways to model proteins that Laura went over. So there are 4 main ways that you can model a protein. Yeah, 4 main ways. And so one of the ways we looked at was ball and stick, which is called wireframe sometimes. The balls represent atoms and the sticks represent the bonds. And with the wireframe, oh, I'm sorry, there's like, it's separate from it. Yeah, so ball and stick model is just like you have, it's like if you're taking organic chemistry, it's just like the student kit. So you have like balls that represent atoms and sticks represent the bonds, whereas the wireframe, you just sort of have wire, which is just like the stick, which shows the bonds, and there's no like balls that are depicted. And then the second kind is the space fill model, which just shows every atom that's within a protein and all the space that it takes up. And these are good because you get to see the overall shape of a protein and like the size of the protein. Then within these models, you can see R group interactions. But a disadvantage of this model is that it's hard to see the backbone of the protein. Another way to model proteins we looked at was the ribbon structure. And these structures usually just show the secondary structure. So you can see the alpha helices and beta sheets in this model, but you cannot see the R groups. You only just see how the path of the backbone is, because remember in secondary structure, it incorporates the bonds of the backbone. And there's not really any, or like R group bonding is not the main type of bonding you see in there. Yeah, so secondary structure really only shows backbone interactions. Then there's a backbone model, which shows just the path of the backbone. You don't see R groups in there. And in this class, I think Laura has an example of models where you can overlay one image over the other. So if you were interested in looking at a particular protein, you could overlay like a ribbon model and a ball and stick model or something, or you can sort of alternate or mix in different models to get an overall shape of a protein. I think she has one of these in her slideshows, which she might show sometime this week, or that might be already in like the lecture slides, but I think she has an example of how you can have two models kind of overlaying each other. And yeah, I have pictures of these models so that you guys can see. And the main idea for these models is just to come up with pros and cons for each, like why one would be good over the other, like whether you'd want to see R groups or just the backbone or overall shape. But yeah, so these are the models I was talking about. And
Okay.
you can see that the space filling, you can see all the atoms that are within a protein, but it's kind of overcrowded. And the ribbon model, you can see the secondary structure, but you can't see the R groups. Did I hear that somebody had a question?
Now, I was just gonna say, can you go back to the last slide for a sec, please? Sorry.
Yep.
Okay, thank you. I got it.
Yeah, so this image, these images are just to go with the last slide. Does anyone have any questions? No? Okay. Chi-Chi, you want to do this slide?
Before I do that, I found a video that like explains the different levels of protein structure.
Okay, we can show that. I just think it will help you guys like to have it more clarified.
Share the
Can
screen.
you send me the link or put it in
Yeah.
so I can play it? Okay. I don't see it.
I just sent it right now.
Oh, thanks. Okay, so I'm gonna play this video for you guys. It's
The
only
shape
2
of
minutes.
proteins.
It's only 2 minutes. It just hopefully can help put those ideas together. And you guys should be able to see it and hear it.
There are 4 different levels of the shape of proteins. There are 4 different levels of protein structure or protein shape: primary, secondary, tertiary, or the 3rd level, and quaternary, or the 4th level. The primary or first level of protein structure is a chain-like or linear sequence of amino acids joined by peptide bonds, as you can see here. The secondary or second level has two shapes, an alpha helix or coil-like formation, or a folded pattern called a beta sheet. In both of these structures, hydrogen bonds between the amino acids stabilize the shape of the protein. The tertiary or third level of protein structure involves more folding and bonding of the secondary structure. The coils may even touch each other as some covalent bonds form from the sharing of electrons between different amino acids. But most of these twists and folds occur from the result of ionic bonds between positively and negatively charged R groups of the amino acids. The quaternary or fourth level is where clusters of more than one polypeptide chain link together to form a giant molecule. Many different types of bonds may be formed within this structure. The shape of protein is important. For instance, fibrous proteins are extended linear proteins that are part of ligaments, tendons, and muscles. And globular proteins fold into almost spherical shapes and have their hydrophobic or water-fearing R groups buried deep within the core, and their hydrophilic or water-loving R groups extended out into the water when in an aqueous environment. Proteins take their shape based on the job they're required to perform, and some have moving parts that are important to their functions. Proteins can have their structure or shape altered by denaturation. When this happens, the protein is no longer able to carry out its job. Denaturation can take place because of change of temperature, change in pH, or the presence of certain hazardous chemicals. And that be the basics.
Okay, so yeah, that video does a good job of just sort of putting the ideas together. But you guys can see that like with the primary structure, it was sort of like zoomed in and you have the sequence, and then as you go on to secondary, tertiary, quaternary, sort of zooms out, but you can see like the primary structure sequence within the secondary, and they just sort of build up on each other. Kind of like I don't know, like Legos, if that makes sense, but like the primary structure, once you have enough of primary structure, you have secondary structure, and then once you have multiple alpha helices, beta sheets, you have the tertiary, and so on and so forth. So hopefully that makes a bit more sense with the video. Thanks, Chi-Chi, for the video, by the way.
Of course.
Yeah, but back to this stuff. You guys can see my screen again?
Yes, ma'am.
Yeah.
Awesome. All right, Chi-Chi, take it away. Domains and subunits.
Okay, so we've already discussed subunits. Subunits are found in the quaternary structure. Like each part of the polypeptide chain is the subunit of that structure. As far as domains are independent. They usually have a distinct function, and you can find the same domains in different proteins, and they have different functions. So that's really the key difference into like domains and subunits. Domains can look the same, but in different proteins, they have different functions. And they're usually independent of whatever it is the protein itself is doing. So they're responsible for particular functions or interaction that contributes to the overall role of the protein. Any questions, comments, thoughts?
So you said it has specific function in a specific kind of protein?
Yeah, so they have a specific function that they do in the protein, which in turn just kind of leads or helps the overall function of the protein in the body. So within the protein, it has a specific function, which contributes to the specific function of that protein, which contributes to the function of the body.
Okay, thanks.
Are domains in like are they in like every structure? Like, are they in like the tertiary structures of most proteins, whereas subunits are only in quaternary structures?
I'm not.
I think subunit is involved in every quaternary structure, because it's like the definition of quaternary structure is when you have two or more polypeptide chains, and like two or more meaning like a group of them, and those like different polypeptide chains is a subunit that contributes to the overall quaternary structure. Whereas domains, I don't know if it's a requirement for every single protein? That I'm unsure of.
So, well, from what I remember, proteins do have a lot of proteins have domains. I'm not sure every single protein has domains, but a
Yeah.
lot of them do have domains, because a lot of the domains, like I said, have a specific function, and each, though they might be similar or though they might be the same domain, they have different functions in different proteins. So, yeah.
Thank you.
Any more questions? Okay. But yeah, like the video said, for proteins, it's really important. Like their shape is really important. So like the form follows the function. So whatever the shape of the protein is can determine its function. So if something goes wrong during like the construction of the protein, like during tertiary structure or secondary structure, like the detrimental effects could be really bad for the protein, so much so that it prevents it from performing its function or doing its job. And you'll learn later on that proteins have different jobs of like breaking down things, synthesizing things, and how like having the correct bonds or the correct sequence of amino acids can be important. I don't know if you guys took biochem at all, or maybe I've heard, but I think sickle cell anemia is caused by just a single amino acid switch in like
Yeah.
protein or something, and that like that one change can cause somebody to have like sickle shaped cells. So you can see how like just switching around an amino acid, like the chemistry of the amino acid or the type of amino acid it is, can have an effect. And yeah. Okay. Any questions? Okay. Yeah, so this is just another diagram of what Chi-Chi just said about the domains versus subunits. We like to throw lots of diagrams at you guys just so you guys have better visual understanding, because some of the ones earlier, like the color scheme of them was kind of off. And I like diagrams, so why not? Yeah, so this is just a summary of domains versus subunits. They are not the same, they're different. So domains basically is when you have like a single polypeptide chain, and it folds independently, and it has like a specific job or function. And then subunits is when you have two or more types of chains, polypeptide chains. So you have several polypeptide chains, or in other words, two or more subunits. And these can create these beautiful 3D structures you see in the diagram to give us quaternary structure. So you can see the difference. I know it's a little tough to understand at first, but this entire thing is one long polypeptide chain, and what the green and the red is telling you is that there are two different domains within the same protein or polypeptide chain. So there are two different domains, and like which confer to different functions of the protein. Whereas in this diagram here, let's look at this one. So with this one, you have 3 subunits: the red, the yellow, the orange, and they combine together to make one mega molecule, one mega protein. So yeah, hopefully that makes some sense. Does anyone have any questions? I'm gonna take that as a no. There's nothing in the chat, Chi-Chi?
No.
And oh, another diagram. Okay. Chi-Chi, do you want to explain it?
Yeah, it's kind of like what we talked about before, the difference between like the tertiary and the quaternary. So the tertiary, like the diagram shows, is kind of just like one person performing the functions, or each like body part does its own function, just like the arms or the legs you're walking. So like think of the arms and the legs are domains. And then for quaternary structure, it's kind of like two different people. They're connected, but they're doing the same thing. So it's kind of like each person is a subunit performing the same tasks. So now, let's say one person is running, the other person is walking. You can see the one person running and the other person, those would be domains because they're doing two different tasks. But in this case, since they're doing the same task, they're subunits of one giant protein polypeptide. So did I confuse anyone with that analogy or anything? Okay, good.
Yeah.
Wait, just to clarify for that too. So when isn't the last one is like each person still has their individually functioning parts, and those are the domains. So the domains are still within like the subunits too, right?
I think yes, because like the subunit is like a polypeptide chain. And then when you have two or more subunits, you have an overall like mega protein structure. But within a subunit, since the subunit is still a protein, within that, you could have like the domains, which is part of the tertiary structure of that particular polypeptide. So it's kind of like zooming in. It's kind of like where is it? I'm trying to look for that hemoglobin. It's kind of like, I was looking for the hemoglobin example because it was clear, but it's kind of like zooming in on one of these and seeing like the tertiary structure of it. It's like zooming in on this and then like it comes up with this, if that made sense. I don't know if I lost you there, Hannah.
No, that makes sense. Thank you so much.
Yeah, of course. I was like a lot of it's kind of tough to see because it's like when you start talking about primary, like you're zoomed all the way in, all the way down to the level of detail of the amino acid. And then as you keep going higher up in the structure, you sort of zoom out. And then that's when you see like the mega structure of the quaternary structure. But yeah, I was hoping that if I find another video that shows that, like shows how it's kind of zooming out, then I'll email it to you guys. But yeah, it's tough to wrap your head around at first, but it will make sense with more like practice and like trying out for questions and stuff.
And also, if you have any further questions that you think of, you can always email us, or also when you do your check-ins, you can also ask that there and we can respond as well.
Thank you.
Yeah, I guess I could try another like analogy with this diagram, but the way I look at it is like let's say the person on the left is me and then the people on the right is me and Chi-Chi. So like me on the left, like I have my lungs, I have my heart, I have my bones, like I have all my different body parts that make up me. I'm one person. Then when I go like hold hands with Chi-Chi, like you kind of like each person is a subunit of like the hand holding chain. So like holding hands is kind of like subunits combining together to make like one big protein, or like in this case like a group combine to make a group of people. But I still like have my own individual body parts, and then Chi-Chi like has her own individual body parts, even though we're grouped together, and like together we could like jump up and down or something or sing and dance, but like uniquely they're separate people. I don't know if that made sense, if I just like confuse people.
No, that definitely made sense. Thank you.
Okay. I always think it's cool to throw analogies in there.
Also think of co-joined twins. That's another way to think.
Sure, okay. That works too. But yeah, I think that's it. I think that's the last slide. That's all we have for you guys today on week 1 content, and Friday we'll cover the rest of the things that are in her videos. And yeah, that's pretty much it for week 1.